Now let me explain a lac operon concept, that is regulation of gene expression in the prokaryote, specifically in E. coli. Okay, we write lac operon concept. Operon concept that comes under the same concept, that is regulation of gene expression. Who proposed it? The two scientists, Jacob and Monod, in a prokaryote, that is E. coli. Okay, I don't remember the year, it might be 1961, I'm not sure about it. Okay, Jacob and Monod proposed lac operon concept. So what is this lac here? Lac is nothing but lactose. Lactose. Okay, we just write lactose. Now to understand this, what I have to do is, the first thing is, let me just give you the diagram of E. coli. Let us imagine this is now E. coli, the bacteria, and this E. coli has the kind of like the DNA which is now naked, okay, naked, and this gene, it has got a lot of genes, and these genes are working because they need to produce a specific type of enzymes or maybe proteins for its survival. Okay, let me just tell you why. Let me consider this particular segment from here till here till here. Okay, from here to here. So let us imagine this is now contains a lot of genes, because if you take any prokaryotic gene, it is nothing but produces so many enzymes or so many proteins from the single strand of single segment of the DNA. So it is called as polycystonic. Okay. So what I am going to do is, let me just write the components of the lac operon. Components. Okay, so what is this operon? I forgot to tell you. Operon is the set of coordinated genes where they perform a specific function in the metabolic pathway. Okay, set of coordinated genes perform a specific function in the metabolic pathway. That is actually the operon here. So that means, in the operon must contain some genes. Many genes should be there in the DNA segment. First, let me just write the components very easy. Let me just write P here, then small I, third one is again P, fourth one is O, fifth one is, let me just write structural genes. Okay, so structural genes. And normally structural genes contains 3 genes, that is polycystonic. Eukaryotic genes are monocystonic. That means one DNA segment contains only one gene, and one gene will produce only one protein or one enzyme. But in this case, one gene will produce multiple proteins or multiple enzymes. So I am talking about lac operon concept. So that means something which is related to lactose metabolism. So the genes which are mainly essential for metabolism of the lactose, they are the structural genes in this case. So let me just write lac Z is a gene, the other gene is lac Y, the other gene is lac A. So we have got 3 genes within the structural gene, so it is polycystonic. Okay, so these Z, Y, A genes are related to metabolism of the lactose. What is this P? P is nothing but promoter. You know what is promoter? Promoter is nothing but, it is the site, it is normally situated in the upstream segment of the DNA where RNA polymerase binds, correct? So you just write that. It is the binding site of RNA polymerase. What is this I? Okay, so I is now, let me just write, it is the regulator gene, the gene which regulates something. Regulatory gene. So this gene will regulate the activity or the function of the structural genes, that is ZYA gene. So regulatory gene is going to block ZYA gene, that is its main function. So regulatory gene, okay. What is this I here? So regulatory gene I can also be, because actually it is based on the conditions. I am talking about lac operon concept. There is one more concept, that is tryptophan operon, arabinose operon, like that. Okay, so here I means inhibitor. So here it's going to be an inhibitor. So it's going to inhibit the activity of structural genes. Or sometimes if you take a tryptophan operon, the same I could be the inducer or like initiator. So it's going to activate structural genes. So the function varies, okay, based on the type of operon concepts. So in this case it is inhibitor. Remember it, I is inhibitor. But for other operon concepts, that is I in the sense it is inducer. Okay, it means it will start the activity of the structural genes. Okay, what is this P here again? The P is nothing but the promoter. So let me just write the promoter. So here the same thing, the RNA polymerase enzyme bind. Okay, a different RNA polymerase enzyme bind to this promoter. Okay, what is this O? O is operator site. O is operator site. What is going to happen in the operator? The product of regulatory gene. Okay, regulatory gene, it is going to produce one protein that is called as repressor protein. So repressor protein is inhibitory protein. It is inhibitory protein which is going to bind to the operator. So it's going to bind to the operator. That is, I can write repressor protein binding site. Yes, the operator is repressor protein binding site. You can easily understand this concept. Okay, now how to understand this? I forgot to mention the functions of LAC ZY and A. This gene, Z gene, produces one enzyme which is called as beta galactosidase. Okay, you have to write all these things in the beginning of the explanation. Very important. Beta galactosidase enzyme. Y actually produces one enzyme called as permease enzyme. What enzyme? Permease enzyme. Third one actually produces another enzyme called as trans-acetylase enzyme. Got it? Beta galactosidase, permease, trans-acetylase. What is the function of beta galactosidase here? Okay, let me just write here. The beta galactosidase will hydrolyze the lactose into glucose and galactose. Okay, remember it. It will hydrolyze lactose into glucose and galactose. Permease actually, which acts like a kind of like a membrane bound, what you call transporter. You know that some proteins will be act like membrane bound. Normally they bound to the plasma membrane and allow the molecules to enter from outside inside the bacteria. Like that, the permease, they give permission for the entry of lactose. So they allow the lactose to enter into the bacterial cell. That is actually the thing you can write. Okay, permease allows the entry of lactose from outside to inside the bacteria. Okay, trans-acetylase. It's going to bring acetylization. Okay, so right here, acetylization of beta galactoside. Okay, so that means it is understood that they are all related to only one type of metabolism, that is breaking down of lactose to produce energy. That is the function of these three enzymes. Okay, all these enzymes are nothing but the product of a single gene. We call it as structural genes. So some segment of the structural gene will produce beta galactosidase. We call it as Z. Some segment Y, or that particular segment Y. A will produce the enzyme trans-acetylase, like that. You need to understand that. Okay, now we will imagine that for the bacteria, which is the natural food, for its survival, it has to take glucose. Okay, so glucose will be now situated outside the bacteria. Okay, it is situated outside the bacteria. It will take the glucose. It's like a food. It will absorb the glucose present in the external medium and it will take it. And it has got some genes. Okay, so let me just consider this gene. Okay, this gene, which is necessary for the production of certain enzymes to break the glucose. That is actually, we know that glycolysis pathway. Glucose, one molecule of glucose, finally, after completion of 10 steps, it's going to produce 2-pyruvic acid, and we know that what's going to happen to this pyruvic acid, which will now liberate 2 ATPs. That means bacteria getting energy. That is from glucose metabolism. Correct? So this is natural. Now suppose instead of glucose, so now glucose is not available. Okay, so what I'm going to do is from the external medium, I am going to remove the glucose. I will remove the glucose. Instead of that, I'm going to put lactose. So I'm just changing the medium. Okay, I will take the bacteria out and put it in a petri plate containing only lactose. So bacteria has to take the lactose, otherwise it will not survive. It has to find a different way by taking the lactose and survive. Got it? So this is the thing. Now here anyhow we have got a gene which is for glycolysis, metabolism of glycolysis. It's going to produce some enzymes for glycolysis, but it is not working now, because there is no use, because there is no glucose inside. lactose is there. That means it has to activate some other genes to break this lactose and to produce the ATP. Yes, that is lac operon. It is the regulation of gene expression, particularly here lactose metabolism. Got it? That is, it has got set of genes. Okay, so let me write here. This segment is called as P. This segment is called as I. This segment is called as P. This segment is called as O. Then here you see the structural genes. That is Z, Y, A. This all these from here till here, whatever the genes you see, they are all coordinated in function for a specific metabolic pathway, that is breaking the lactose. So the lactose will enter and lactose will be converted into different form, because lactose is a disaccharide, it has to be converted into monosaccharide. Got it? So finally ATP is synthesized. That is actually lac operon. So now we have to understand the two cases of the lac operon concept. That is case 1, when lactose is not available, glucose is present. The second case is glucose is not available but lactose is present. Two cases we need to study to understand the regulation of gene expression that is related to lactose metabolism in E. coli. So let me erase everything now. Now let me write case number 1. Case 1. Okay, when glucose is present. So glucose is the natural food for the E. coli. So we write glucose is present. Okay, so when glucose is present, I do not want the structural genes to be active. I need to shut the structural genes from working. Got it? Okay, so what I want to do is, let me just write lac operon is switched off. You need to write this. I do not want the structural genes, that is Z, Y, A, from and producing that the beta galactosidase, permease and trans-acetylase enzyme. So I need to stop it because lactose is not present, only glucose is there. There is no need of activating these structural genes. Got it? So I need to stop the structural genes from working or from producing these different enzymes. So lac operon is now switched off when the glucose is present. Okay, to understand that, let us just draw one diagram. And let us consider this, the double stranded DNA of the E. coli. Okay, so let me write here DS DNA, double stranded DNA of the E. coli, and let me just use a red color. So within a specific sequence of the DNA, there is one structure which is called as promoter, normally situated in the upstream region. Okay, so this is the now promoter P. And now I will use the pink color. Very close to the promoter, there is another gene. There is one gene called as gene I. The gene I in the sense regulatory gene. So you know what is this gene I? Regulatory gene. Here I for lac operon concept. Lac operon concept, I is inhibitor. For tryptophan operon, I is initiator, that is inducer. Are you getting my point? Okay. Now what I am going to do is, like here, somewhere else here, let me use the blue color. I put one more box and I will put P here. This is promoter. So it is understood that this is the promoter site for the gene I. Got it? That means RNA polymerase will bind to this P site, that is promoter site, and it will transcribe the gene I. Okay, it's going to transcribe gene I. Whereas this promoter is for structural genes. Okay, that means it is understood that for a specific gene there is always a specific promoter site. I will not mention the terminator site, not required. Okay, so P here, and let me just write O. I think better I use the O. Okay, this color. This is now O. O is the operator region for the promoter for the structural genes. And let me use the purple color here, Z, Y, E. And let me just put the blocks, put them in the blocks. Here let me write structural genes. So we have 3 structural genes, hence E. coli prokaryotes have polycystronic genes. Got it? So this contains maybe a single gene or single DNA segment can produce multiple proteins. That is structural genes, that is what you call the polycystronic genes. Okay, fine. So let us just assume that this is now the promoter where the RNA polymerase now binds, and here, okay, I forgot to mention. Let us assume that the entire board is now the cytoplasm of the E. coli where the glucose is available. So that is glucose is present. Okay, so let me just put the glucose is now present, available. So I do not want ZYE from being producing the different enzymes to break the lactose, because glucose is available. Okay, which is a natural food for the E. coli. So RNA polymerase now binds to the promoter and it will transcribe gene I. So that is the regulator gene. It's going to produce one mRNA. The name of the mRNA is repressor mRNA. What do you call? Repressor mRNA, which is produced from regulatory gene, and this repressor mRNA is now going to encounter one ribosome and it's going to produce a polypeptide chain, that is nothing but a protein, okay, during translation mechanism. So let me use the purple color itself. So it's going to produce one protein. I'm using purple color. Okay, so this protein is called as repressor protein. Repressor protein. Here I would like to add one point for your competitive examination. The gene I, if you take many genes, okay, so many genes are there, but the gene I, which is now regulatory gene, which is active 24/7, which is active throughout, which is going to produce the repressor protein all the time in all the prokaryotes. This is very, very important. Hence it is called as constitutive genes. You may get a question. Give an example for constitutive gene. Constitutive gene is nothing but gene I, that is regulatory gene, which is active throughout. Okay. So now this repressor protein is now produced from the regulatory gene, and the tendency of the repressor protein is it's always, once it is produced, it's going to bind to the operator side of the structural gene. It's like that. It's like it is going to bind. So let us imagine there is another RNA polymerase which is going to transcribe ZYA to produce the mRNA for different enzymes, which will now bind to the promoter, and now it has to move right to this side, but we have already noticed that the repressor protein is now bound to the operator side because it is the tendency. So let me just mark like this so it will be easy for you. Got it? So it is blocking the path of RNA polymerase. It's like an obstacle for RNA polymerase. So RNA polymerase now cannot enter the structural genes. So what's going to happen now? The entire structural gene is now switched off. It's not going to produce any enzymes. So Z will produce beta galactosidase, Y will produce permease, A will produce transacetylase, but in this case it is now switched off. Why? The RNA polymerase cannot read the structural gene, that is template DNA of the structural genes, cannot get transcribed to produce the mRNA. So these genes, the enzymes are not synthesized. Are you getting my point? Yes. So this is lack of operon is switched off when the glucose is present. So it's like it is not necessary for ZYA to be activated when glucose is present, because they get activated only lactose is present when glucose is absent. So this condition is called as lack of operon switched off. So entire thing is now switched off, but this segment is switched off. Got it? So this is the thing. This is case number 1. Let us see case number 2. Let me erase it. Now let me write a case 2. Case 2, that is when glucose is absent. Okay. So that is lack operon is switched on. Operon is switched on. Let me write here. Switched on. That means I need to activate ZYA genes to produce the different enzymes for the metabolism of lactose, because glucose is absent. So let me write here, glucose is absent. Glucose is absent. Instead of glucose, what is present? Lactose is now available. Okay. So lactose is not the natural food for the E. coli. So again, I am going to draw the same diagram. So it is understood that in the examination you need to draw 2 diagrams to explain lack operon concept. Very, very important concept. Normally you get this question. Okay, so I don't know, I forgot which color I used for the different segment. Let me just write P here, I here, then here we write P, O, Z, Y, E. How do you read it? Piposia. It's easy. Just remember this Piposia. That is actually the reading. So it will be easy for you in the examination. You will not forget the arrangement of these different parts of the lac operon model. Okay, so let us just put the compartments. Pipo, Okay, so this is O now, Z, Y, A, that is structural genes. Okay, so now we know that this RNA polymerase will bind to the promoter and it will transcribe the gene I, that is regulatory gene, and it's going to produce the mRNA. What do you call that mRNA? Repressor mRNA. Same, everything is same, and it's going to be translated to produce a protein. So let me use the purple color now. The protein is repressor protein. R protein. Repressor protein is the product of regulatory gene. Okay, it has got the tendency to bind with the operator and bind and blocks the path of RNA polymerase. Okay. Now let us imagine the lactose is now trying to get the entry into this cytoplasm of the bacteria, because you know the lactose is not present now. But remember that for the activation of this lac operon, that is, I need to switch on this lac operon, I need at least small amounts of lactose to be inside the cytoplasm. Otherwise, it's not going to activate these ZYA genes. So let us imagine that, let us take like 1 or 2. So these dots are nothing but lactose. At least let us imagine minimal amount of lactose is present within the bacteria. Let me tell you how exactly lactose is present. But I said like lactose is absent earlier. Now lactose is present inside. Only 2 are there. Very minimal amounts of lactose is there. So this lactose is acting as an inducer. Okay, you may get a question. Name the inducer of lac operon concept. Inducer is nothing but lactose itself. Even though lactose is undergoing metabolism process, but still it is an inducer. Why it is called as an inducer? Because it's going to switch on the lac operon, that is these genes. Got it? Because it's going to bind to this repressor protein. So lactose is now disaccharide which is now going to bind to the repressor protein. See, when it binds, it's going to modify the structure of the repressor protein. Modified. Structure is modified. So when the structure is modified, it cannot bind to the operator. Structure is modified. It cannot specifically bind to the operator because it's modified. Structure is now changed. Okay, it's like we can say denaturated. So what's going to happen? For the promoter, RNA polymerase enzyme now binds and now there is no blockage in the path of RNA polymerase. So it's going to read the template strand of the structural gene where ZYA genes are now transcribed to produce the mRNA. So let me just give you the diagram of mRNA here. Okay, so this mRNA is the part of gene I, gene Z, and this is the part of gene Y, and this is now gene A, like that, like mRNA codons. And it's going to produce, this Z will produce beta galactosidase. No need of writing full name, right? Beta galactosidase. Y will produce the permease enzyme, and A will produce the transacetylase enzyme. That's it. So all this now, okay, let me tell you what is this permease. As soon as the permease enzyme is produced, it's going so very close to the plasma membrane. Let us imagine this is the plasma membrane of the E. coli, and it going to act like a transmembrane protein where it allows more. I have used blue color for lactose. Lactose is outside now. Yes, it will allow more lactose to enter, get the entry into the cytoplasm. See, got it? So you know it, the main function of permease enzyme. So what's going to happen? The beta galactosidase enzyme will now encounter each lactose and break them, that is hydrolyze them into glucose and galactose. Got it? Okay. Whereas transacetylase will bring the acetylization of this beta galactoside, like that. So it has got different specific function. And finally ATP is synthesized. ATP is now synthesized. That's what like the bacteria is getting energy. So this is now when the glucose is absent but lactose is present, the lac operon should be switched on. I need to switch on these ZYA genes. Hope you understood this. You need to write all this stuff. Very easy. Okay. So that's all about it now. Let me just continue for the NEET students. So for board students it's enough. Okay. Hope you understood this.